So, what do we call the male gametophyte in case of flowering plants? We call it as androecium. Now, the next part we are going to see the gynoecium part. Gynoecium, it is called the female part, that the female reproductive organ of the flowering plants. Now, this gynoecium, what does it contain? It contains an ovary, then style and then a stigma, right? So, this is how a gynoecium looks. This gynoecium is also called as pistils or carpels. Now, you are going to see some of the pictures of these carpels. Since these carpels, when they are in multicarpellary means there is more number of carpels, more than 2, right? So, you can see in this figure, it shows that the carpels are fused. When the carpels are fused, they are called as syncarpus. Example is papaver. And the next one, the next picture you can see, it is of the micalia. What you can see? These carpels are free, okay? This multicarpellary of the carpels, when they are not fused, okay, when they are free, we call it as apocarpus. So, this multicarpellary of the pistils can be of 2 types. It can be either apocarpus, syncarp. If you look at this figure, it is the structure of angiospermic ovules and we call it as anatropous ovule. So, what is this funicle? This funicle is a stalk to attach to the placenta of the ovules, right? So, this is the funicle and the next one is hilum. This hilum is the junction of the body which attach with the funicle, right? That is called the hilum. And the next part, as you can see here, is integuments. These integuments, you can see outer integuments and inner integuments. These are going to form the seed coat of the ovary. And the next one is micropylan. This micropylan, it is a small pore in which the integuments are not enclosed. And the next one is chalazolan. This chalazolan is in the basal part of the ovule. So, it represents the basal part and it remains opposite to the micropylar end. And the next one is called as the nucellus. Nucellus, it is rich in food reserve and it is present inside the integuments. These integuments are enclosing this nucellus. And inside this nucellus is the embryo sacs. This embryo sac is the main female gametophyte. Now, we are going to discuss about the megasporogenesis. What is it? Megasporogenesis, that means the formation of megaspores. And what is megaspore? Megaspore is the female spores, right? Female cells. So, what happens? See, this megaspore formation will take place with the help of megaspore cells. And these megaspore cells are residing at the micropylar end of the ovules, as you can see in this figure. So, what happens? This megaspore contain dense cytoplasms and a nucleus, right? Now, in the second figure, what happens? This nucleus are divided. How it divided? It divided through mitotic division. Because of this mitotic division, the nucleus in the megaspores divide and it divide into dyad cell. This is the dyad cells you can see here. And the next is the tetrad formation. So, these mitotic cell divisions will form 2 cells. These 2 cells will divide again to form 4 cells. They are forming megaspore tetrad. Remember that microspore tetrad and megaspore tetrad. Microspore tetrad will form when the pollen germination will take place and the megaspore tetrad will form when the female gametophytic development will take place. So, in this megaspore mother cells, 3 of the cells will get degenerate and 1 become functional. During this 1 functional, what happens? There will be some changes which take place inside this functional megaspore cells. This megaspore, what it going to do? It going to divide, the free nuclear division. There will be only the division of the nuclear. There will be no cell formations, right? So, the first divisions you can see is of the 2 nucleus and this again it forms 4 nucleus and again it forms an 8 nucleus. So, what you can see here? There is a 8 nucleus. Now, this 8 nucleus is a free nuclear division. There is no formation of the cells. And the later stage after this 8 nuclear division, they are going to form a cells. Wow, how many types of cells they will form? 7 cells. So, we call them as 7-celled 8 nucleate. So, these 7 cells, what is going to do? 3 of the cells will move to the chalazolan and that is called as the antipodal cells. And the next 3 will move it to the micropylar end and that is called the egg apparatus. It contains 1 egg cells and then 2 synergid cells and the last one is filiform apparatus. The function of filiform apparatus is to guide the pollen tube so that it can enter the egg cells, right? So, that is called the egg apparatus. That egg apparatus contains what? The synergids and egg cells, right? That is called the egg apparatus. And the next 2 nucleus will move it to the central part of the embryo, and that will be called as the polar nuclei. So this is how it looks, okay. This is the embryo 6 of the female gametophyte. What you can see here? There is a 3 cells, 3 and then 3, 6 and then 1 whole gametophyte is containing 1. So what it forming? It has 7 cells, and how many nucleus have? 1, 2, 3, 4, 5, 6, 7, 8. So 8 nucleus, right? So we call them 8 nucleus and 7 cell female gametophyte. It is very clear with you. Now what will be the ploidy of this female gametophyte? Obviously it is going to be haploid in number. Why it is going to be haploid in number? Because the megaspore mother cells are dividing meiotically. After meiotically, what is the ploidy? The ploidy becomes half. It becomes haploid, right? This kind of development of the female gametophyte is called as the monosporic development. Now, can I ask you what is monosporic development? Monosporic means the development is taking place with 1 megaspore cells, right? And the 3 will become degenerate. So this is how it looks. So this is the overall embryo sac. So this is about the female gametophyte. And the next is Urey and Miller experiment. Miller created similar conditions of temperature and pressure in laboratory scale. Let's see this figure to understand the Miller and Urey experiment. So this is the diagrammatic representations of Miller's experiment. So at the first time he has done the circulations of the 4 types of gases. These 4 types of gases like the methane, the ammonia, hydrogens and water vapors. And it was filled in a airtight apparatus, you can see here, and passed electrical discharge from the electrodes at 800 degrees centigrade. And then he passed this mixture through a condenser, you can see in this figure, and he circulated the gas continuously in this way for 1 week, and then analyzed the chemical compositions of the liquid inside this apparatus, and he found a large number of simple organic compounds including some amino acids such as alanine, then glycine, aspartic acid, and thus he proved that organic compounds were the basis of the life. So you can see the boiling water has been carried out through the mixture of gases which contain this methane, ammonia, hydrogen and water vapor, and then it was allowed to pass through the condensers, and the water contained the organic compounds, and after tasting these organic compounds he has observed that it contains certain kinds of amino acids like alanine, then glycines and the aspartic acids. Similar kind of experiments were done by some other scientists to analyze the components like sugar, nitrogen bases, pigments and fats. And then analysis of meteorite content also reveals similar compounds, which reveals similar processes are occurring elsewhere in the space. These experimental evidence about the origin of life is called as the chemical evolution of life. And the next, what do you mean by abiogenesis? Abiogenesis is the first form of life arose slowly through evolutionary forces from non-living molecules. So with this, let's start with the second topic of this chapter, that is evolutions of life forms, a theory.